Hi, everyone. So I'm Kristy Townsend. I'm a co-founder and chief scientific officer at Neurite Inc., where our goal is to make neuropathy right. So what is neuropathy? Peripheral neuropathy is a medical condition where peripheral nerves die back, so we lose proper tissue innervation and control of tissue functions from the brain. The top cause is diabetes or prediabetes. So if we think of the experience of a typical patient, they might have diabetes and start to experience some unexplained symptoms. So things like numbness or pain, burning and tingling in their lower legs. And then they go to see their primary care doctor to try and figure out what's going on. And here's really the critical window, because if they don't get an accurate diagnosis at this point, then worse disease outcomes will happen. So this is where in the clinic, there are currently not very good diagnostics for small fiber neuropathy. And this is a progressive and degenerative condition that can eventually even lead to limb amputation if not caught early enough. But with an accurate diagnosis, the patient is not only aware of their condition, but now we have better disease management, earlier implementation of treatments, and adherence to lifestyle modifications that can improve their quality of life, because the peripheral nerves can regenerate if their neuropathy is diagnosed quickly and early. So looking at the problem more closely, it's caused by over 30 different medical conditions. I told you the top is diabetes, but other top causes are also aging, chemotherapy, HIV, and now COVID-19, which is a growing cause. And these are all small fiber peripheral neuropathies, which are tough to diagnose. So over 100 million people in the US alone have diabetes and prediabetes. So just in this market, it's estimated that up to 70% will eventually develop peripheral neuropathy. The costs are high to patients, to medical payers, and also indirect costs due to loss of productivity. The global diabetic neuropathy market was valued in 2021 at 4.3 billion. And because there's currently no reliable and accurate diagnosis of small fiber peripheral neuropathy, for 30 million peripheral neuropathy patients in the US, this is billions in preventable costs. So a quick background on the difference between large and small fiber neuropathies. So large fiber neuropathies affect major nerves, such as the sciatic nerve that runs down the back of the leg. And right now there's good functional diagnostic tests for that type of neuropathy. Small fiber neuropathy affects the tiny axons that are innervating our tissues and organs. This is the most common, as I mentioned before, and also the most difficult to diagnose. So in talking to clinicians and patients, we know there's a consensus need for a functional quantitative test for small fiber peripheral neuropathy that is fast, easy to use, and relatively pain-free. Small fiber neuropathy is unique because for patients, it's a really long road to diagnosis right now. We're essentially trying to diagnose a small fiber condition using large fiber tests. It's progressive and degenerative, so an early and sensitive diagnosis enables better treatment approaches. And we've learned all this through over 100 customer discovery interviews we've done as a company through an NSF I-Corps program, and also 100 individual patient interviews we did to understand more the patient perspective. So our solution at Neurite is a device that allows more sensitive detection through peripheral nerve recordings through the skin and subdermal tissues. Our MVP device we call the DEN, or the Detecting Early Neuropathy. This is the diagnostic portion of what is a patent pending theragnostic platform. It's a hollow microneedle electrode array device that sits in the skin where needles penetrate down to 4 microns, and they can measure reduced or altered electrical signatures along the disease progression of neuropathy. So if you look on the left here, this is healthy tissue where the skin and the subdermal layers are fully innervated by the peripheral axons. And as the disease progresses and the nerves begin to die back, you first lose skin innervation, and then later on lose innervation of deeper tissue layers that can even begin to affect some of your internal organs. Right now, all of the tools in the clinic that are used for neuropathy diagnosis are fraught with very high false negative and false positive rates for small fiber. The only two functional tests, as I mentioned, are EMG and nerve conduction velocity, which are specialized for large fiber neuropathy. And if we look at the total available market for PN diagnostics at 388 million, and then narrow in on the SAM just for the NCV and EMG tests, and then take a conservative 1% of that, we think our achievable market here is 2.4 million. The DEN has advantages to all of the existing diagnostics in use in clinics currently. So focusing in on the two gold standard diagnostics, so skin biopsy right now is still the gold standard for small fiber, and then those two large fiber functional tests. We have the problem that the skin biopsy is good for small fiber, but is not a functional assessment, and these large fiber tests do not accurately diagnose small fiber neuropathy, but they are functional. So our product really improves upon both of those existing standard of care tests. In addition, our product is low cost. It has the potential to deliver treatments, and it can be done really in any clinic setting. So there's no need for specialized personnel. We've demonstrated the differentiators of our device, both in mouse and human studies so far. We've completed a preclinical test in a diabetic mouse model, and we found through our proprietary machine learning that we have over 90% accuracy in predicting which animals have neuropathy or no neuropathy. And this paper is now posted as a preprint online. We've also been validating a new flexible design of the array. So now with a flexible PC board, we're able to mold this to the skin surface. So here's an example showing a human lower leg where the device is applied and just held in place with a simple ACE bandage. And we confirmed in these early human validation measurements that we, similar to mouse, can detect both small and large fiber nerve activity in humans. So here's an example where we asked the participant to flex their muscle, and we're able to pick up large fiber EMG signatures in addition to the small fiber signatures. So now our goal is to move this into a first-in-human study comparing patients with diabetes versus healthy controls. So our current capabilities of our MVP device are to detect neuropathy, to screen and to diagnose with disease. And also as more treatments are becoming available, and they are on the horizon, many of them, we can also measure the regeneration of these nerves and monitor recovery. And because we're a theragnostic platform, we also have future capabilities, so delivering new treatments and detecting biomarkers. And our goal is really to provide an at-home monitoring option similar to a blood pressure cuff or a glucometer, so patients at high risk could monitor themselves at home. But in total, this device offers an earlier and more accurate diagnosis of small fiber neuropathy, saving clinicians and patients time and money, and improving disease outcomes by allowing an earlier window for therapeutic interventions. So our story as a company was that this invention really came out of an academic research need to measure small fiber nerve activity. And then we realized there was also a societal and clinical need. So we founded Neurite in 2018 as a spin out from my prior university, University of Maine. We last year completed a phase 1 NSF STTR, completing those preclinical tests I showed you, and our IP was filed in 2021. We now have been manufacturing our human flexible device, and our first-in-human study will be underway this year, looking at patients with diabetes. To date, we've had over $500,000 in non-dilutive funding, and now we're targeting additional grants and VC funding for our future clinical trials. My co-founder, Magda Blaskovich, and I are the two neurobiologists that run the company. We also have a business lead, Derek Houtman, and really expert consultants, including our co-inventor, who's a microneedle fabrication expert, Rosemary Smith, and an electrophysiologist, Len Cass. And we have an incredibly talented team of other people that I don't have time to go through, but we have neurologists on our clinical team and also consulting on our scientific advisory board. We have computational and electrical engineering consultants and a business advisory board. And together with this team, we've really been able to have good momentum on our business timeline. So where we are currently, as you can see with the green arrow, is in this phase, starting our first in human study and building the human diagnostic algorithm. We have an IRB underway for that right now. And then next we'd move into phase 1 and 2 clinical trials to provide data for the FDA, where we hope to obtain breakthrough designation. So our IRB is under review for this first-in-human study to optimize the device recordings in humans with diabetes and develop that human-specific diagnostic algorithm using non-dilutive funding. And our ask for all of you today is whether there are any advisors or potential mentors who can help us shape our go-to-market strategy. So who will be the key decision makers for this product? Who is our first customer, et cetera. So thank you very much for your attention, and I'm happy to take any questions.